We're hearing today from Kyle Apley from WashU. St. Louis is developing insulin variants to target insulin-binding B cells in type 1 diabetes. Dr. Apley is a research assistant professor in the Department of Biomedical Engineering at WashU St. Louis. As a protein scientist and chemist, his research is focused on developing protein-based therapies to suppress undesirable immune responses in autoimmune disease that have desirable qualities for clinical translation. He works in collaboration with engineers and immunologists to design, synthesize, and evaluate protein therapeutics in T1D models, and he earned a PhD degree from the Department of Pharmaceutical Chemistry at the University of Kansas as an NSF graduate fellow in 2022. This paper is evaluation of proinsulin Th1nk as a targeting ligand for insulin-binding B cell in autoimmune diabetes. I'll drop that link into the chat. And also, if people can ask questions at the end, we'll have the chat going and you can drop a question in real time and we'll field them at the end. Thank you so much for joining us, Kyle. This is really exciting and a lot of people in the room interested in hearing more.
Thanks, Monica. All right. Hi, everyone. Thanks for joining me today. Today, I'm going to be presenting on a recent publication evaluating an insulin variant to try and target insulin-binding B cells in autoimmune diabetes. Here you can see my list of co-authors, but I want to especially thank the collaborating labs that are part of this project. I'm under Corey Birkeland in the Birkeland lab at WashU, and we're really thrilled to have both Dr. Rachel Bonamy at Vanderbilt and Dr. Peggy Kendall here at WashU work with us on this project and help us explore some ways to hopefully rein in the autoimmune response in type 1 diabetes. To start out, before I get into the paper, I just want to take a little time to speak about some of our motivation for this work and what we're hoping to achieve. First off, I don't want to belabor a lot of the type 1 diabetes background, as I know this audience is probably already quite interested in type 1 diabetes. But for us, what we're most interested in exploring is how we can take advantage of insulin-binding B cells acting as antigen-presenting cells to try and halt the autoimmune response in type 1 diabetes. So, there's a growing body of work demonstrating that there are a sizable number of B cells that infiltrate the islets in humans in type 1 diabetes, and especially this is overrepresented in the younger onset disease cases. And from working in the NOD mouse model, we're able to understand that insulin-specific B cells, as a subset of these islet-infiltrating B cells, specifically are helping to sustain the autoimmune response locally there in the pancreas. And so our understanding is if we can shut down this antigen presentation pathway or make it more tolerogenic in nature, we may have the capabilities to kind of selectively dampen the autoimmune response against the islets. So we're also very interested in kind of precision medicine, I guess, or using specific and very safe immune modulating approaches. Specifically for type 1 diabetes, we are interested in trying to advance antigen administration approaches since they are more safe, have less off-target effects. And if we're thinking about ways that we could safely try and prevent disease progression in young adults and juveniles, antigen administration approaches could be very suitable if we can get the desired efficacy. So I'm sure a lot of us are familiar that there have been a slew of studies investigating the administration of islet antigens like insulin, for example, in mouse models and even in clinical trials. And unfortunately, there's been quite minimal benefit, maybe some responders in very specific subsets in humans, but the broad efficacy that we're hoping for hasn't been there. I believe that we can increase the efficacy of antigen administration approaches if we enhance them in some way. Specifically, that might be, increased targeting to tolerogenic sites where presentation by some of these tolerogenic APC subsets could be strengthened. Alternatively, since we're fighting an ongoing autoimmune response, it may be necessary for the delivery system itself with the antigen to have some sort of tolerogenic context to drive that tolerogenic message along with the antigen. And alternatively, There's also strategies that people have proposed to try and delete these antigen-specific B cells that may be promoting disease in an antigen-specific manner. Yeah, and so there's been a flurry of activity in this kind of space, especially over the past decade, as we've seen a lot of progression in, for example, the antibody drug conjugate space, where in cancer they've been able to demonstrate the ability to have improved selectivity with delivering active compounds that either promote cell death or, like with some of the more recent glucocorticoid modulator drugs that have been under investigation as ADC payloads, potentially to suppress rogue autoimmune responses in a selective manner. And some of our collaborating groups have also shown that tolerization or elimination of insulin-binding B cells in the NOD mouse model does protect against diabetes. And so we're interested in further investigating that approach. Now, the roadblock that we encountered when we were first interested in pursuing this work a number of years ago is that there are quite a few challenges with using insulin to try and target these insulin-specific B cells in vivo. And really, if we're trying to think about doing work now that has the potential to readily translate to the clinic, we need to go ahead and address these roadblocks as early as possible to make sure they don't put up a fight later on. So specifically, insulin, as we know, is hormonally active, and it doesn't take much to result in hypoglycemia. So first off, it is absolutely necessary that these insulins not be hormonally active. But further beyond that, we also want to limit off-target binding as much as possible for a couple reasons. Insulin in the body is rapidly endocytosed by cells that have high expression of the insulin receptors, such as the hepatocytes. And so if we're thinking about trying to maximize antigen exposure, if that's the route we're trying to achieve, we really need that PK profile to be as long as possible and the half-life in circulation to be as long as possible to support that. And this degradation and uptake by hepatocytes and other insulin receptor expressing cells would negatively affect that. Additionally, say we're using this insulin as a targeting ligand to try and deliver some sort of payload to these insulin-specific B cells. Uptake of these payloads, whether that's something like a steroid or more of a cytotoxic compound or even something like that, if that is taken up substantial quantities by these off-target cells, we could see a lot of undesirable toxicity. Lastly, from more of a pharmaceutical perspective, the stability of this insulin molecule needs to be suitable for eventual formulation, such that a pharmaceutical product could be prepared that would be stable on the shelf or in your fridge for a sufficient length of time, such that patients could easily self-administer this compound or have it readily administered in a clinic. And insulin, say if you're administering it right now because you're diabetic and you're administering typical insulin, it's typically stabilized in these zinc hexamers. And that's because insulin in its monomeric state tends to try and form these fibrils, basically these stacked beta sheets that are insoluble precipitates. And up to this point, a lot of pharmaceutical preparations of insulin has been formulated to stabilize them in the hexamers. However, you could imagine that if we start attaching payloads to this insulin, or it's in some sort of maybe particulate system, we couldn't necessarily rely on hexamer formation to keep that protein stable in the vial. So therefore, we need to try and find an insulin variant that is more stable to physical means of degradation. Also, insulin does self-associate. So if we find ourselves working with a multiply valent system, for example, self-association of these insulin molecules could negatively affect function. So these challenges I shared with you aren't necessarily new. These challenges have been understood for a long time. Here I have two patents, one of them from George Eisenbarth in the mid-90s and the other from a team at Novo Nordisk shortly thereafter. And at this point in time, multiple people had hypothesized that administering these non-hormonally active insulin variants could potentially be a curative solution for type 1 diabetes by allowing us to administer more and more insulin which had previously been limited due to its hormonal activity. And these are both variants that we took into strong consideration. One of them over here on the left is desoctapeptide insulin, where residues B23 through 30 are removed. Those are the ones shown in yellow. Another one is this Th1nk insulin, where the B25 phenylalanine is mutated to N-aspartic acid. And now both of these do a great job at reducing hormonal activity. However, desoctapeptide insulin is notoriously unstable from a physical standpoint, even less so than insulin. So that is one reason why we felt like there wasn't a clear answer of what to use to move forward. Another option besides these two is native proinsulin, which is as the protein is expressed after the prepeptide's cleaved in the body. It has the C peptide indicated in yellow. And this C peptide, if you leave it on, does improve its physical stability and provides a slight decrease in hormonal activity. However, the amount of decrease in hormonal activity is not sufficient on its own. So therefore, with these three molecules as inspiration, we set out to perform the studies described in this paper to try and determine, can we combine some of these aspects of these molecules to find an insulin variant that is well suited for potential future use as a targeting ligand to target insulin-specific B cells. And with that, beyond the hormonal activity, off-target binding, and stability, we also wanted to better understand how well does our envisioned insulin variant bind to these anti-insulin antibodies. And part of that is later on here, the study will show how Rachel Bonamy's team panned for anti-insulin B cells in pre-symptomatic T1D subjects and what we found from the B cells that are isolated in that work. Okay, so again, our work that I'll describe here, the goals we had again were try and identify an insulin variant with negligible binding to the insulin receptor and activity, have increased physical stability, and then also retain binding affinity to these anti-insulin B cells that we're trying to use this insulin variant as a targeting ligand for. So first off on this slide, I want to take a minute and just talk a little bit more about the structure of insulin and proinsulin on the right side. That way everyone follow along and better understand the different changes that are being made. So up there in the top right is the key. The orange spirals denote what sections of proinsulin exist in alpha helical structure. I thought that's helpful in case you're a structural biologist that's really familiar with the structure of insulin. That might help you kind of serve as landmarks when you're looking at this two-dimensional structure. Additionally, there are 3 disulfide bonds holding insulin's core structure together. Those are represented with the black dotted lines. And then within insulin itself, the dark yellow sphere indicates the F25D or the B25 residue, which is mutated from phenylalanine to aspartic acid, and the F25D mutation. And then the light yellow halos identify the amino acids that are mutated to try and reduce self-association. Lastly, in an effort to make this insulin variant more easy to use in terms of synthesizing these constructs that are using this as a targeting ligand and then some sort of payload attached, we used a sortase motif on the C terminus of the protein to allow site-specific conjugation. And we elected to do that at this stage of the project because we wanted to minimize the chance that attaching molecules elsewhere on the protein might block either activity or binding to B cells. So the species we evaluated as controls, we had insulin and desoctapeptide insulin, and then we evaluated insulin with the sortase tag, insulin F25D with sortase tag, native human proinsulin, proinsulin with the sortase tag, and then proinsulin with F25D mutation and the sortase tag, and then species 8 and 9 have a couple additional mutations to help reduce self-association. I won't spend much time on those during the talk today. In short, we didn't see much change in function or binding due to the LISPRO or DKP mutations. So first up is exploring how the combination of adding the C peptide and the F25D mutation affect hormonal activity and insulin receptor B binding affinity. Again, don't want to cause hypoglycemia, and we also want to limit both how quickly these insulin variants are taken up by hepatocytes, for example, and potentially how much off-target delivery there could be to hepatocytes. And this was investigated for hormonal activity using a reporter cell line, and then insulin receptor B binding affinity was evaluated in a radioisotope study using HepG2 cells and iodine 125 insulin in a competitive binding format with the insulin variants. So starting with the hormonal activity, we could hardly see an increase in the reporter cell line output with the proinsulin F25D, even at 100 micromole concentration. So very, very low hormonal activity. And this was even, at least, maybe 10 to 50 times lower than what we observed with insulin F25D. And as you can see here, looking at the changes or the change in outcome with the proinsulin versus the insulin 25D, it seems like retaining both the C peptide and the F25D mutation result in an additive decrease in hormonal activity. So we were really thrilled to see that. Additionally, with the insulin receptor B binding affinity, we see pretty similar trend here. Retaining the C peptide and the F25D mutation resulted in the greatest reduction in insulin receptor B binding affinity. Under these assay conditions, we didn't see more than 10% binding of the insulin variant, even at 1 micromole concentration. And so this also was a great improvement over insulin F25D and proinsulin on its own. So from this work so far, we're pretty enthusiastic about proinsulin F25D and its ability to more selectively target these insulin binding B cells versus other cells in the body. Next up, we evaluated the physical stability of the insulin variants by doing a fibrillation assay. So like I mentioned earlier, insulin, when it's in its monomeric state, kind of likes to aggregate and form these fibrils. Typically, it's stabilized in these zinc hexamers, but I don't think we can be confident that we'll be able to use that strategy to formulate these enhanced insulin compounds in the future. To evaluate stability, we used a Th1nk fibrillation assay. This is pretty typical assay for looking at fibril formation. I will say that this assay was performed under accelerated conditions. So this is like pH 2, I think 60 degrees Celsius, so quite harsh conditions. There's some other work out there that evaluates fibril formation at more physiological conditions, pH 7.5, 37 C, and the amount of time it takes to do those studies is just incredibly long. And there does seem to be some correlation between fibrillation rate under accelerated conditions and physiological conditions. So that is one limitation of our work. But we were very encouraged to see that the proinsulin F25D achieved about a 50-fold longer fibrillation time versus insulin. And as you can see here, desoctapeptide insulin is even more prone to fibril formation than insulin itself. So definitely, in looking back to some of the options that were available early on, the proinsulin F25D definitely gives a clear advantage over desoctapeptide insulin in terms of the data we've seen so far. Additionally, just over here on the right, chart of dynamic light scattering data to look at the particle size of these insulin variants. And I just included this here to show that we see a larger hydrodynamic radius for proinsulin and proinsulin F25D that don't possess the LISPro and DKP mutations, which affect self-association, to just demonstrate that those mutations did perform as expected. Under these accelerated conditions, all of the insulin variants are forced into a monomeric form due to the pH. So that's why we don't really see a difference in terms of fibril formation rate between the LISPro and DKP and the regular proinsulin. Okay, so now with some more of the biophysical characterization out of the way, let's turn our focus to evaluating how these insulin variants bind different anti-insulin antibodies. So we really have, I guess, two sources of importance for these anti-insulin antibodies that we can work with. One of them are those that are derived from animals. For example, from immunizing a mouse with human insulin, seeing what anti-insulin antibodies are generated. And one of those antibodies that we found to be of interest is MAB125, which is isolated from the hybridoma cell line AE9D6. And the reason why we're interested in this one, even though it is mouse-derived, is it is a very high affinity antibody. The binding affinity for insulin is like 1 to 2 nanomolar. And there seems to be a high similarity in the binding epitope for MAB125, along with high affinity human-derived anti-insulin antibodies from polyclonal sera. So therefore, we felt like this animal-derived antibody would be a good surrogate to at least help us probe how the insulin variants might bind to a single high-affinity insulin antibody. Additionally, our collaborators, specifically Peggy Kendall's lab and her past mentor, Thomas's lab, had evaluated models of non-obese, or the NOD model, that are transgenic or have a fraction of their B cells express this MAB125. And as you see in the chart here at the lower left, in the mice that express a low level of this MAB125 B cells, you have an increased incidence of diabetes compared to wild type NOD. And down at the bottom, if you do the same transgenic transformation, but use an antibody that's not specific for insulin, you get a reduced incidence of disease. So we have some understanding that this antibody and its specificity, even though it was initially mouse-derived, does have some pathogenic potential in autoimmune diabetes. So we evaluated the binding of all our insulin variants to this MAB125, and we found that there was no substantial differences in binding affinity against all the variants tested. We evaluated this using 2 different experimental techniques. We used biolayer interferometry, and then also a plate-based method that I will call an equilibrium ELISA. This equilibrium ELISA, there's the reference there. It's an older method. Essentially, you equilibrate the insulin or your antigen and the antibody in a non-binding plate, and then you use a direct ELISA to quantify how much unbound antibody is present. And from that, you can essentially calculate, the amount of free antibody present and do a binding curve that allows you to calculate KD. Both of these approaches gave us pretty much identical results. All of them showed a binding constant, or a KD, of less than 10 nanomolar. And due to the small size of insulin, on BLI, it gives a pretty weak signal, as you can see by the intensity of the noise in the charts on the right. So as we moved forward to working with human-derived antibodies, we chose to keep working with the equilibrium ELISA. Okay, so next up is working towards isolating insulin-binding B cells from pre-symptomatic T1D individuals. This work was done by Rachel Bonamy's lab, along with her collaborators at the Vanderbilt Medical Center. Here's just a quick kind of overview of the process they used. Patients were identified through the TrialNet pathway to prevention. Patients that were accepted into our study group had to be positive for, I believe, 2 or more autoantibodies, but were not yet symptomatic, so they're not yet taking exogenous insulin. PBMCs were then collected, and then the PBMCs were stimulated in a plate in order to drive B cell antibody secretion. And then each of these wells was screened by ELISA to identify wells that contain insulin-binding antibodies, meaning the presence of an insulin-binding B cell. Then hybridomas were formed from those B cells, and the hybridomas were either used to express this anti-insulin antibody that we're going to use as a surrogate to tell us, can these insulin variants bind to these insulin-binding B cells? And then later on, the hybridomas, the IgG genes were sequenced to enable recombinant expression of these various human-derived antibodies. And Dr. Bonamy's group also has a great paper explaining kind of the methods and approach that they use for isolating antigen-specific B cells in various disease states as well. That's indicated here at the bottom of the screen if you're interested in learning more about her process. From all the PBMCs tested, there were 7 positive hits in the insulin binding ELISA coming directly from the plates. And so of these 7 that were first identified, we tried all of them in the equilibrium ELISA, first with insulin to confirm if these antibodies could bind insulin in solution. Of the 7 that we evaluated, one of them, T1nk, indicated over here on the right, demonstrated that it could bind insulin in solution at a concentration that was within the feasibility of our technique. So then we went ahead with T1nk and tested it with all of our insulin variants. And as you can see on the table here and the graph on the left of the screen, MAB T1nk did not bind any of the pro-insulin species, only insulin and insulin Th1nk and the sortase tagged insulin. It also did not bind desoctapeptide insulin. So just from the epitope mapping that we can do based on the variants tested, it appears that MAB T1nk binds somewhere near the C terminus of the B chain on insulin. So let's see here. Again, MAB 125 is understood to bind where the light gray circles are. So this is known as like the A chain loop epitope, A8 through A10 to A13, depending on what source you're using, and potentially B1 through B3. T1nk appears to bind at the C terminus of the B chain, potentially here at this proline, lysine, and threonine from B28 through B30. We're pretty confident in that due to the fact that retention of the C peptide blocks binding, as well as if we delete residues B23 through B30, there is also no binding. So from this, it was interesting to see that this lower binding affinity antibody, which for insulin, the binding affinity was about 1 to 2 micromolar, so about 100 to 1,000-fold weaker than the MAB 125. However, it still had reasonably high specificity for insulin. Yeah, but unfortunately, it did not bind our pro-insulin Th1nk species. So with that, I just want to go over a few quick conclusions, then we can do some acknowledgements and then open up time for questions. So from this work that we did here, we found that pro-insulin Th1nk is a suitable insulin variant to incorporate into antigen-specific tolerance-promoting strategies. If you're working on evaluating those in non-models of disease that are enriched with MAB 125 expressing B cells. If you're curious at seeing how administering your antigen administration strategy in vivo affects a specific population of B cells, pro-insulin Th1nk would be a great model antigen to use because there are already transgenic lines of not available where either 1 to 3% or 90 to 95% of the B cells express the MAB 125 BCR. And this is a line that we've used in some adjacent studies and some work that's ongoing but not yet published to see how we directly impact a subset of B cells, specifically ones that are insulin specific in the mice. Furthermore, pro-insulin Th1nk does have more desirable pharmaceutical properties than DOP insulin and insulin Th1nk, but we'll have to wait for further studies to help us further understand the contribution of these T1nk-like B cells to disease to help clarify if insulin Th1nk or would pro-insulin Th1nk be more suitable for clinical development of some of these antigen administration strategies. So with that, I'd like to thank some people that have made this work possible, specifically within the Berkeley lab at WashU and previously at KU before we joined WashU a couple years ago. Jalen, Grant, and Kristen, as well Rachel Bonamy's group. We're thankful to have the assistance of our graduate student Lindsay Bass, as well as some other folks there at Vanderbilt. And then Peggy Kendall's lab as well. And although not co-authors on this paper, both Amber and she have been immensely helpful in some other projects we have ongoing in collaboration with them. Additionally, thank you to the Type 1 Diabetes TrialNet Study Group's Pathway to Prevention study for support and allowing us to investigate some of these patient samples in this work. And lastly, I'd like to thank for funding the Helmsley Charitable Trust and a number of fellowship programs for supporting trainees. and lastly, the National Institute of Health's various funding programs for support as well. And lastly, before I open the floor for questions, I'd just like to say that we underwent this work to try and identify a suitable insulin variant to enable us to explore targeting these insulin-specific B cells in vivo in some of these mouse models I've briefly mentioned. And we published this work and really wanted to share this to let people know that if you have a platform or technology that you're curious about evaluating in type 1 diabetes, or maybe you're already exploring platforms of type 1 diabetes but haven't been able to explore your platform with insulin due to the challenges we discussed, we think that proinsulin F25D or one of these similar variants could be a great way for you to be able to explore your strategy and the potential for type 1 diabetes. And it would be great to connect and see if there's a way that we can work together to evaluate your ideas.
That's great. That's a fantastic collaborative spirit. Thank you very much for adding that. I wanted to open it up for questions. Please unmute yourself or raise your hand or drop something in the chat. Matthew Schaefer, are there particular epitopes on insulin that are commonly bound in T1D patients? Or do different patients have different epitope binding antibodies?
Yeah, that's a great question. And that's something that I think Dr. Bonamy's group's working on actively to try and better understand. A lot of prior studies, to my understanding, looking at B cell specificity and in more general autoantibody specificity for insulin have been limited to testing polyclonal serum from these patients. So essentially it'd be like running an ELISA and just seeing, for the different species of insulin, do you observe the same responses or not? And so a lot of the epitope specificity and epitope mapping there has also been limited to just the differences in insulin amongst different animal species. So for example, you go look back at some of the older papers and they'll be using fish insulin, cow insulin, pork insulin, several other species just to try and figure out maybe where these antibodies bind.
Yeah, that's really all over the place.
Right. And one thing that I really appreciated Rachel trying to understand in this paper is the anti-insulin antibody repertoire doesn't necessarily exactly reflect the epitope specificity of the B cell repertoire in type 1 diabetics that might actually be functioning as antigen presenting cells. Because, for example, it's kind of understood that the antibodies themselves aren't that pathogenic, except maybe helping with antigen uptake by antigen presenting cells through FC receptors. So, we're really hypothesizing that it's these other B cells that can function as APCs themselves that are really kind of helping drive disease locally in the tissue. And it's hard to directly probe that due to the challenges with acquiring pancreatic samples. And so, for this study, we did have to rely on samples from PBMCs, but I think that still helps us understand, or at least get closer to understanding what this B cell repertoire looks like.
Yeah, that's going to be challenging. Is there anyone else who has a question? Vikas?
Yeah, hey Kyle, wonderful talk. I just have one question. I'm just curious to know, looking back at the structure of this proinsulin, like the amino
Yeah.
acid sequence, did anybody try to mutate these cysteine residues in terms of, gaining the functional activity in terms of pharmaceutical activity? Because I can see two cysteine are very close.
Yeah,
So
so
yeah.
the interface of insulin that binds with the hormonal insulin receptor is, if you're looking at the insulin 2D projection on the page, let's say you're looking at the front of it, it's more so the back that interacts
Ah, I
with the
see.
hormonal insulin receptor. For example, the B24 through B26, that's kind of referred to as the aromatic triad, because that's kind of an anchoring region of insulin that interacts with the hormonal insulin receptor. There are definitely other amino acids that can also be mutated to reduce hormonal activity. When we picked this F25D mutation, we did that based off of looking at where we thought these B cell epitopes might be and trying to avoid placing a mutation within one of those. We do have some other unpublished data where we tried another mutation to reduce antibody binding
Mhm.
or not antibody, hormonal activity, but it ended up destabilizing a whole region of the insulin. Even though it was quite far away from one of the antibody binding epitopes, it also greatly reduced antibody binding as well. So yeah, it's
Yeah, I was just wondering about, if you can mutate these cysteine residues and then you prevent, the aggregate or like, multimer formations. Yeah, that's
yeah,
what I was
oh,
thinking.
I see. Yeah, there have
Yeah.
also been studies on adding and subtracting cysteine residues to insulin to try and improve stability.
Mm-hmm.
I can't recall exactly, but I believe that, if you have, as you reduce the amount of cysteines, you definitely destabilize the molecule. And I think that would make it a lot more prone to either fibril formation or other
I see.
physical
Mhm.
degradation mechanisms.
Okay. Yeah, thank you.
Alrighty, anyone else?
I just was gonna say, I think Kyle did a good job answering the first question. It's a good question, and it's one that I wish we had an answer to, but that's an area of active investigation. I think he did a pretty good job summarizing it. I guess the only other like point I might make is we have a question about, people have clearly seen in the polyclonal sera that you can find these high affinity responses to insulin, and there's some evidence to suggest that you can find high affinity B cell receptor responses for insulin too. But I will say that in the mouse, even this antibody that he was mentioning has high affinity for human insulin. It does not have high affinity for its autoantigen, rodent or mouse insulin. And for the most part, at least we have not captured in the studies we've done so far any high affinity human anti-insulin B cells either. And there was a rather old study where they had done immunizations of BALB/c mice. So they, have normal enough immune tolerance mechanisms intact, you would think, and they used foreign insulins on purpose and of course adjuvants to try and drive the system. And you really don't get high affinity antibodies out, at least from that one, what I'll call case study, as far as the self, or in that case, mouse insulin is concerned. And so, we have this question of whether these high affinity B cells have much to do with initiating the disease, or whether this is just something that maybe after, years of beta cell attack, maybe eventually you start to get these things, but whether or not that's what's happening in the beginning, I think we just don't know. And that's something that we're thinking a lot about. And one of the questions that Lindsay Bass, a really fantastic grad student in my group that Carol already mentioned, is, trying to parse out now. But it's something that we're certainly thinking about. And I think it's important when you're considering how these T cells and B cells are first coming together and driving the response. TrialNet is a perfect example, and there are a few other organizations out there that are allowing us now, in clinical trials to actually intervene at much earlier stages than, even 5 or 10 years ago was just not feasible, meaning, before diabetes onset. And as we start to be able to intervene at these earlier stages, I think knowing how this initiates is really important when you think about the types of strategies that are going to, either succeed or fail, and perhaps even within, dare I say it, specific endotypes of type 1 diabetes, which I'm a proponent of that concept as well.
Yeah, well said. Can you just comment, just as sort of my last question was, this whole idea of sort of the smoldering autoimmunity and then a second hit, how might what we're talking about here be, if you would, activated, like in that scenario?
The short answer again is we don't entirely know, but we have some educated guesses. So John Williams is a PhD scientist who's actually still working some with my group. He, in his PhD work, had shown that if you take an anti-insulin B cell from these mice that Kyle was mentioning, these MAB125 B cells, if you stimulate them with insulin, they don't proliferate very well. But if you give them LPS and engage TLR4, now you can drive proliferation. Some more recent work by a postdoc in my group, Dudley McNitt, showed it followed up on some of John's initial observations that if you take T-dependent immunization, so in this case, we're talking about either sheep red blood cells conjugated to insulin or CFA, insulin emulsions, classic T-dependent immunogens, you do not drive a very good anti-insulin antibody response via these T-dependent immunizations. You can interestingly push the anti-insulin B cells to enter the germinal centers and adopt that phenotype, but there's something that's holding them back from differentiating into antibody secreting cells. Contrast that with this, what we like to call a T-independent immunization, acknowledging that this isn't perfect, but there's this kind of weird cocktail that Tom Thomas had been studying for a little while. There's this Brucellus ring test antigen, Brucellus abortus being the bacteria. It's actually the antigen, but it's, got a cocktail of some TLR agonists in there. When you hit them with this T-independent immunization, they actually make anatomical germinal centers. That's why I say T-independent with an asterisk. A mouse that doesn't have a thymus can mount a response to this antigen. So that's why we categorize this T-independent. This can make robust, fairly quick germinal centers, and actually they make boatloads of anti-insulin antibodies. So there are clearly settings, Monica, to your point, there are clearly settings where whatever this immune tolerance checkpoint is, that's at least restraining them from becoming antibody secreting cells and maybe causing premature death in the germinal centers. This is something we don't quite know yet. You can release this if you, hit them with the right thing. And so then this concept of, did you have the misfortune of having a certain viral infection or, something like that, or some kind of bacterial infection? I mean, there's a lot of data from other labs that I think show some compelling evidence that there are certain triggers that are associated with type 1 diabetes. And I think some of this, trying to parse out the exact timing, is of course tough in humans, easier in mice, but ideally we have studies in both settings that start to help us get a leg up on what exactly is, or probably a few things, right? There's probably not one type of immune tolerance breach and one, bad bug, right? There's probably a bunch of things that, depending on the individual, might be enough to push you to the not just having enough autoreactive T and B cell clones, but actually driving them to become these, kind of more effector-like populations, meaning for the T cells, they're starting to make pro-inflammatory cytokines. For the B cells, they're starting to have these, co-stimulatory molecules and things that are upregulated that are allowing them to now act as, great APCs, which is exactly what we don't want them to do.
Yeah, many different ways to release that break. I would just also wanted to say it's excellent seeing the entrance of new labs, Kyle's lab joining the Bonamy lab, also Stephanie Hanna over in Cardiff are just shining a light on sort of what seems to be underappreciated B cell activity in this disease state. So it's really great, amazing. And both the Bonamy lab and the Hanna lab are contributors to the T cell, B cell data repository. And so thanking them also for that participation. Very important to kind of understand the landscape here, and B cell is a part of it. So thank you so much for everything you're doing in the space. Thank you, Kyle. And it was a great talk, really interesting and thought-provoking. And hope everyone has a great rest of the day and a great 4th if you're in the US.
Thank you all.